A federal committee in the U.S. has given its approval for gene editing trials in humans. The trials will be the first to be carried out in humans and will help provide data on the safety of a new technique, which has been shown to be successful in animals. An advisory committee for the National Institutes of Health approved the use of CRISPR, a precise gene editing tool borrowed from bacteria, to boost the effectiveness of existing cancer treatments. Researchers hope the technique, which enables precise editing of DNA, will eventually lead to the widespread use of treatments which use a patient's own immune cells to target cancers. The trials will reportedly be funded by a newly established immunotherapy foundation, which was set up by former Facebook president Sean Parker, who provided $250 million of funding for the project. Parker previously called the research a Manhattan Project for curing cancer with the immune system. However, while the approach may have the green light from the NIH, trials will need the approval of the Federal Drug Administration before they can go ahead. CRISPR is being looked to by biomedical scientists as the future of genetic-based therapies. Since its discovery in bacteria, it has become a widely used tool for genetic engineering. The technique involves a DNA cutting enzyme and a small tag which tells the enzyme where to cut. By editing this tag, scientists are able to target the enzyme to specific regions of DNA and make precise cuts, wherever they like. It is hoped that the approach will lead to targeted therapies to treat genetic conditions, with cancer being the ultimate prize, using the body's immune system to target and kill cancer. Despite the high hopes for CRISPR to aid cancer therapy, the approach is yet to be proven safe in humans. However, early clinical trials using similar forms of gene editing augmented therapies have been carried out. In the proposed trials, a total of 18 patients with different cancers will have T cells removed and editing using CRISPR. These immune cells recognize and target foreign invaders, killing them before they can spread. Researchers hope that by tweaking their DNA, the T cells will recognize the rogue cancer cells as a threat. According to Nature, the major concern will be the potential for unwanted edits to the patient's DNA, where the tool could recognize another part of the genome as a target, snipping the DNA and potentially leading to faulty repair, mutations and immune problems.